Good morning, everybody, and welcome to UCD's Institute of Food and Health Research Bites. So our speaker this morning is Dr. Colm Collins. So he's no stranger to UCD. His undergraduate was in pharmacology from UCD, and then he went on to do a PhD in immunology. And then following some time as a postdoctoral fellow here, he moved for quite a number of years to the University of Colorado in Denver. So Colm then returned to Dublin as an ad astra, so we're delighted to welcome him back, and it's I suppose a very prestigious appointment in terms of the caliber of people we're looking for under the ad astra scheme. So congratulations to Colm on achieving that. So in terms of his research, it looks on the development of novel therapeutic approaches for the treatment of intestinal inflammation. So I'll hand over to you, Colm, at this point.
Sure.
If you want to share some of your research ideas with us. Thanks very much.
Absolutely. Yeah, thanks very much. And thanks for the invitation today. So what am I, today I'm going to talk a little bit in case what I'm talking about, or what on earth is he talking about? I think it's often the question that comes up. So I'm going to talk a little bit about career in terms of how I came to be here and what really drives me in terms of what I like to research and where it all kind of came from. I'm going to talk a little bit about past experience in terms of, what successes we've had based on this our approach to research, and then from there to move into where we've moved to now. So where our focus is and how we're directing our research at the moment. And then, as I mentioned, some of the issues that we have in terms of patients and their needs, I'll talk a little bit about some of the future directions that our research is moving into, because we're obviously keen to address some of the shortfalls in our current research pipeline. But just by way of introduction, I started here in UCD. I think everybody goes to university to experiment with drugs, and I was no different. So I did my BS in pharmacology, and from there I knew immediately I kind of was bitten right away, and I went straight without any rest or break into a PhD with Professor Alan Baird and Deirdre Campion in the vet school so not a million miles away. And I loved it. I really enjoyed it. But then, went straight from that into a postdoc with Tars and John, and I think that was really kind of fomented my interest and highlighted some areas that I thought were really interesting. So it was a nutraceutical based project. We were focused on the role of beta glucans in porcine intestinal immunity. And I realized a couple of things there. I was fascinated with this notion that we could take endogenous or naturally occurring products and have a protective or a prone or anti-inflammatory effect in the gut. And I also knew that I wanted to stay in academic research, and that I kind of loved the academic environment. I found it, it was constantly changing and constantly exciting, and it was something that I wanted to do. But it also meant that I wanted to go somewhere else to get experience, so that I could come back to UCD and then, use that experience that I'd gained elsewhere. And so based on that, then I decided to move, and I went to, having lived at home for 28 years, I decided enough was enough. And I went to Colorado, which is probably fairly far away. But I moved to the University of Colorado in Denver. It's beautiful, 300 days of sunshine. And while all this was going on, obviously, life was happening as well. So there was a very attractive postgraduate in the lab in the vet school. She followed me to Colorado. She eventually agreed to be my wife, and we had a couple of kids. But at that point, we really realized that we wanted to be back home, and we wanted to be around family. We wanted to be in Ireland, and most of all, we wanted to get back to UCD, where we'd realized we were kind of at our, we were really keen to be involved. And so it's tough. We said we'd go for 2 years to the States. And 12 years later, we're still kind of thinking, we probably should think about moving back home. But when there's two of you, it's a little bit more challenging. And so we were really lucky when the ad astra program opened up, and there was a couple of permanent positions opening up in pharmacology and physiology at the same time. And so I applied, and I was lucky enough to get an ad astra and then come back to UCD and back to pharmacology. So basically, I got the job in June, sold my house in July, moved back to Ireland in August, and started work in September. And it was going really, really well right up until the whole COVID situation. So it's been an interesting 12 to 24 months, I think. But during the course of my time in the US, my research was focused on inflammatory bowel disease. And this is actually, it's a bit of a misnomer. It's not a single disease, it's a spectrum. So we have Crohn's disease at kind of one end of the spectrum, which is a transmural disease, which affects anywhere along the GI tract. But it's also transmural in that it's affect the epithelial layer on the surface of the gut, but it penetrates then all the way down into the muscle layers. And that contrasts with then ulcerative colitis, which is a very much more superficial disease. It typically starts at the rectosigmoid region and it progresses retrograde back up along the colon and up towards the small intestine. And so it's these kind of two diseases that we focus on. And it's a huge issue here in Ireland. So, you know, European wise, we have about 3 million people who have inflammatory bowel disease. But in Ireland, our rate's about 21 in every 100,000. So it's not horrendous. But we also have the highest increasing rate of incidences in the developing nations over the last 10 years. And that's not hugely surprising because we know that as a country develops economically, the rates of IBD increase significantly, and it's associated with economic development. We get this kind of rise in inflammatory bowel disease, and we're seeing it here more so than we're seeing it anywhere else at the moment, and particularly in adolescents and in pediatric IBD, which are kind of the worst, most kind of difficult to treat areas for IBD. And so, in terms of challenges associated with inflammatory bowel disease, there's a couple that are looming large at the moment. So, you know, there's been incredible progress made in the last 10 years in terms of therapeutic development. We've seen a lot of new antibody-based therapies come online, like anti-TNFs that are really effective. But even the most effective of those, which is Remicade or infliximab, it's effective in about 70% of patients initially, but in most cases, or in many cases, patients lose any kind of efficacy within 12 months because they develop anti-drug antibodies, and then they can clear the drug quite quickly. And as a result, the drug becomes completely ineffective and the patients need to be cycled on to a new therapy. At the same time, because of the heterogeneity of patients who have IBD, there's a huge spectrum. And so it's very hard to get a good therapy that works for every patient. And so in most cases, in the best case scenario, something like infliximab or vedolizumab is only effective in about 20% of patients because there's such a variety of different disease phenotypes. And as a result, you know, there's a huge shortfall in terms of therapies available. And our goal has been over the last 10 to 15 years to try and add to the number of different possible alternative options for drug development. Because I think whereas academics are more inclined to develop completely distinct pathways for anti-inflammatories, the drug or the pharmaceutical industry have made great strides in modifying existing therapies. But our niche is much more in the new types of alternative therapeutic approaches. And then another issue that we've highlighted, that's been highlighted for us recently, is that because all our newer therapies the majority of our new therapies are antibody based, we start to run into problems where the current animal models that we have can't adequately predict efficacy of human specific antibodies. And so we're in a very awkward situation where we've had, you know, horrifying circumstances like with natalizumab or Tesabri, where there was an unpredictable off-target effect, and it couldn't have been picked up in a mouse modeling system. And so some of the newer therapies like vedolizumab, which is an anti-integrin antibody, it's completely ineffective in rodents. And so it becomes very hard to study drugs like that. And the newer drugs that are being developed are very difficult to study in an animal model. So in terms of the disease, then the pathogenesis of the disease is kind of, it varies with patient, but there's a certain level of commonality. So the hypothesis we have now is that patients develop some sort of initial triggering event, whether it's stress or anxiety or smoking or an environmental pollutant or a bacterial infection or a viral infection or brain damage or, you know, alcohol, all these different things can trigger a transient gut permeability disruption. And that allows bacteria then to cross the gut barrier and they trigger an immune response where dendritic cells that are that should normally trigger a non-inflammatory or a pro-regulatory response, they instead drive in a pathogenic T cell immune response. And in a specific subset of patients who are people who have genetic predisposition, they go on to develop this chronic relapsing and recurring disease where activated T cells then stimulate more cytokine release that triggers increased leukocyte recruitment to the gut that causes infiltration, tissue damage, injury, further recruitment, loss of barrier function, edema within the tissue, and all of this kind of self-perpetuating cycle, which eventually results in the development of memory T cells. And even when the disease does resolve, those T cells will circulate away. They can then return to the gut 6 to 12 to 24 months later and initiate a second wave of inflammation, so that patients develop this kind of relapsing and remitting disease. And so what our goal has been to develop therapies in and around this kind of area. So we've tried a number of different ways of doing that, whether it was targeting the dendritic cell end of things with drugs like retinoic acid and FLT3 ligand, targeting T cell education with things like cholesterol, or targeting endogenous anti-inflammatory pathways. So things like lactoferrin or alpha-1 antitrypsin. And even targeting the microbiome. So using specific probiotic bacteria like Bacteroides cellulose lyticus, we can suppress the inflammatory immune response and have a kind of protective effect there. And to the kind of higher end of things, looking at specific microRNA with an antagomer or a microRNA inhibitor, blocking a specific microRNA which was involved in IL-10 production and could be enhanced or could be suppressed to enhance a kind of pro-regulatory T cell environment. And really what it came down to was that certainly the ones I was most proud of, I think, led on to patenting with lactoferrin and alpha-1 antitrypsin, were basically just modifying existing endogenous anti-inflammatory pathways. And so that kind of theme stuck with me. And we started to work on a separate endogenous anti-inflammatory pathway. And in this case, it was the cannabinoid pathway, or the endocannabinoid system. And it was kind of a good timing in Colorado because they were just going through legalization. There was a lot of interest and a lot of demand. About a third of our pediatric patients were using cannabis with their parents' help to treat their symptoms, because cannabis can actually suppress GI motility. It can suppress a certain amount of visceral pain, but we really don't understand enough about how it does work. So we think classically of the phytocannabinoids like THC and cannabidiol, but we also produce our own endogenous anti-inflammatory endocannabinoids, so anandamide and 2-AG, and these then bind to specific receptors. And in this case, we're focusing on the CD4 T cells. So we're focusing on the CB2 receptor because that's the only one that they express. And it's handy because it's non-psychoactive, so it's a useful drug target. And so because of the patient's interest in it, there were a number of clinical trials looking to take advantage of the kind of GI motility effects of cannabinoids. But what they found overall is that patients certainly felt better if they took a lot of cannabis. They didn't necessarily perform better. So their empirical outcomes, so measures of CRP or some sort of clinical lab-based testing for improvement in IBD patients, there was absolutely no improvement whatsoever. And in fact, there's a 5-fold increased surgical risk associated with cannabis use in terms of for IBD patients, which is very much the worst case scenario for an IBD patient to require surgery. At the same time, there was a study looking at rhesus macaques that showed that if you give THC, that you can actually promote gut homing of CD4 T cells. And while that might be protective in the context of HIV, in the context of inflammatory bowel disease, that's obviously very detrimental. We want to avoid encouraging cells into the gut wherever possible. But it was this gut homing effect that we were really interested in. And so we took this to our animal model. So this is a TNF-driven model where it basically it's a modification of TNF. So it's overstabilized the gene. It produces enormously high levels of TNF, and it develops a spontaneous chronic small intestinal inflammation. And as you can see here, if you block the receptor with an inverse agonist, then you can attenuate disease significantly. But this taught us a number of things. We were able to publish this, and that was great. And we could show a clear anti-inflammatory effect of the drug. But really, we don't understand an awful lot, A, about the drug, and B, about what the receptor itself does. And so in order to kind of get at this, I performed what's called not a efficient experiment, obviously, an unbiased screen using single-cell RNA sequencing. So we sequenced about 20,000 different cells from wild type and inflamed mice. And we looked specifically for this gene CNR2 that encodes the cannabinoid receptor 2. And we said, okay, well, what is unique to cells that express this receptor? And we generated a list of different genes that are specifically associated with cannabinoid receptor expression. And then while this was expensive but quick, the next step was very much slower, was the conventional PCR to validate some of those target genes that we saw that were differentially regulated. And we were lucky here in that we were able to take advantage of a tool that we're developing, which was based on a gift from our colleague. That was a GFP reporter strain of mice that they express GFP everywhere that they express cannabinoid receptor. So we can actually start to pull out those specific cells and sort them by a flow cytometry and identify those cells in particular. And using this tool, we can actually take it a step further because it uses the LOXP system, so we can actually cross this reporter strain of mice with different specific Cree strains like the CD4 Cree, FOXP3 Cree, Villain Cree, which is an epithelial marker. And we can now generate mice that are specifically deleted for each of these, in each of these target tissues for that cannabinoid CB2 receptor, and start to look at how that impacts inflammation. But I mentioned briefly that there was an impact on gut homing, and that was to do with the integrin beta-7 expression. So integrins form these heterodimeric pairs, and alpha-4 beta-7 is the one that we're predominantly interested in the gut because it's specifically associated with gut homing. And so drugs like Tisabri, natalizumab targets alpha-4, but not specifically, and hence has off-target effects. Vedolizumab, which is an alpha-4 beta-7 antagonist or inhibitor, is far more specific and is quite effective in IBD. And what we found using our GFP reporter strain is that the cells that express that cannabinoid receptor are much more likely to be gut homing. So we think that there's an association between gut homing receptors and cannabinoid receptors. And we can demonstrate this in a functional assay as well. So if we co-culture our cells, treat them with retinoic acid, which we know drives gut homing, or with JWH133, which is a CB2 agonist, or with GP1A, which is a CB2 inverse agonist or inhibitor, we can see the same effect. So if we stimulate the receptor with JWH133, then we can increase gut adherence, so interactions between alpha-4 beta-7 and its cognate ligand MADCAM1. We can drive that and increase it. And we know that that would increase diapedesis of cells across that endothelial barrier then into the gut. And equally, we can do this in vivo. So if we take cells in vivo, when we say, well, we have our GFP reporter cells, we fluorescently label two different cell types. So we've got fluorescently labeled cells that lack the receptor versus fluorescently labeled cells that express the receptor. We mix those together and then we co-inject those cells into a lymphopenic recipient mouse. So a mouse that has no T and B cells. And then we look 24 hours later and see, well, okay, we put the cells in. We know that if they're there, we know that we were the ones who put them there. And where did they get to? And so if you look at this, you can see that in cells that are Cree positive, in other words, that they lack the receptor, we get a significant decrease in homing of cells to the gut, specifically into the ileum. We get a reduction in cells then moving on into the draining lymph nodes and the mesenteric lymph node particularly. And obviously, because the cells have to go somewhere, if they're not going to accumulate in the gut, then they're going to be present in the spleen instead, because what's happened is that they've remained in circulation. They haven't been able to cross that intestinal barrier, and so they're stuck in the spleen and in the peripheral blood. And this is really promising because we know if we can do that, based on the results of what we've seen with other clinical studies, that if we can prevent cells from trafficking to the gut, then we can prevent a certain amount of that intestinal inflammation. And so we took this then a step further and we said, okay, well, we generated mice that were deficient in the receptor and also co-expressed that TNF delta ARE disease-associated mutation. And in cells that had, or in mice that had the receptor present, we saw at 20 weeks of age, mice develop this chronic, severe intestinal inflammatory response. And you can see it there on the left-hand side. There's an enormous lymphocytic aggregate. They develop tertiary lymphoid organs or aggregates within the gut. So the cells don't even need to traffic out of the gut to become educated, and they can proliferate, and you get an incredible expansion of that T cell compartment. You lose any kind of tissue architecture, so the gut's not able to absorb nutrition anymore because the nice long villus-like projections that the small intestine would normally have are completely obliterated. And as a result, you're left with this damaged tissue But if those mice then lack the cannabinoid receptor, and specifically on their T cells, so we're not knocking it out anywhere else. This certainly isn't a whole body deficiency. This is a uniquely T cell specific deficiency, and in those mice we get a significant reduction in inflammation, and more importantly, it looks like the cells that we're losing out on are those pathogenic memory T cells, which are associated with the relapsing and remitting nature of the disease. So really promising findings. And so then where do we go from there? Well, the first thing we know is that we have a limitation. We clearly aren't able to test some of the newer therapies that we have with the current mouse model systems that we have. And so one way that we're looking to address that is we've started developing what's called a humanized mouse. And I'll explain that in a second. Another area that we're moving into is to look at that GI motility aspect, because we know that post-surgical anesthesia associated drop off in GI motility is a real problem in patients as well as in animals. And so looking to use cannabinoids to modify that motility issue is kind of another area that we're looking to branch out into. And then obviously zebrafish, we're moving into a zebrafish model because we're looking for different models to start to understand gut health and gut well-being. And the zebrafish is a really good model in which to do that. So I'll just finish with showing you some of this humanized mouse model and how it works. We basically have a mouse that's so immunodeficient that it has no effective immune response. So it's a combination of multiple different immunodeficient mice. And it's called the NSG mouse. And what it allows you to do is engraft human immune systems. So in our case, we take PBMCs from our IBD patients, engraft them into the mice, and then within a week, they'll have engrafted. And we can start challenging those mice with repeated ethanol enemas until eventually they'll develop spontaneous colitis. And that colitis is populated by human immune cells. So now we can study human immune function, but just happens to be in a mouse rather than in patients, because obviously patients don't appreciate ethanol enemas that often. So with that, I'll just finish by saying I'm looking at, I have funding from the Crohn's and Colitis Foundation, as well as the Ad Astra Fellowship Program. And much like the rest of the country, I think I'm currently applying for SFI funding. So fingers crossed.
That's great, Colm. Thanks for